Hello everyone. So in this session we will discuss the protein metabolism, right? So last time we discussed about the RNA metabolism, and also we have discussed before that we discussed the carbohydrate and DNA metabolism, right? So in this session we will talk about the protein synthesis and yeah, right, and how it is getting matured, right? So that we will talk about. So we will first start with the protein synthesis, right? And this protein synthesis step process is called as the translation, right? So there is a hypothesis called adapter hypothesis. So initially people they didn't used to know about the how this translation process actually works, how the proteins get synthesized in the cell, right? So at that time the amino acids were known, and it was known that protein is made up of a chain of amino acids, right? And it was also known that from RNA proteins are being made, right? So now who is the mediator between the mRNA mature, messenger RNA and amino acids, right? That was not known, right? So for that, uh, Crick actually given a adapter hypothesis where he said that there can be existence of an adapter, right? Which suppose for example this is the codon for a particular amino acid, so probably uh, that adapter has complementarity with this codon, right? So that's why it can bind and it can carry one amino acid. So that's how these amino acids are uh, getting synthesized, or right, or getting converted into the polypeptide chain. But now we know that this adapter is nothing but tRNA or transfer RNA, right? So that actually carries amino acids and that can bind with this mRNA to the particular specific codon through its anticodon loop. So that we will discuss. And the organelle where this translation and protein synthesis takes place, that is ribosome, right? So yeah, so in the rough endoplasmic reticulums, right, in the cell, uh, so in the cell membrane of this, in the surface of this rough endoplasmic reticulum, there are accumulation of ribosomes, right? So these ribosomes are the which is a RNA protein complex that actually plays a role in protein synthesis or translation, right? So this is an electron micrograph of ribosome sitting on the rough endoplasmic reticulum. This is your cytosol, and this is the rough endoplasmic reticulum over which this black small dot, those are the ribosomes. They are sitting over it, right? So yeah, so that is called the adapter. So the adapter hypothesis, and this is the electron micrograph of ribosome sitting on the rough endoplasmic reticulum. So we talked about codon here. So codon is the part of three. Codon is a triplet of three, right? Nucleotide, which is a nucleotide triplet which can code for a particular amino acid, right? That is called codon. For example, ACU is coding for a particular amino acid. So for for every specific codon there is a particular amino acid. So basically the tRNA that will interact with this codon, right? And then according to the tRNA which can interact with this particular codon, they can carry a specific amino acid. So those amino acids can be incorporated into the growing polypeptide chain. So this is amino acid codon chart, right? So you can see that this is a that this chart has a information to tell us that uh, each the all possible nucleotide triplets which acts as a codon, and what are they encoding. So you can see that, for example, if you start with phenylalanine, then uh, there are yeah, so there are two codons, UUU and UUC. They are the codons which can actually encode for phenylalanine, right? So if there is UU, yeah. So if there is UU, yeah. So if there is UUU or UUC, then they will interact with the tRNA, which can carry the phenylalanine, right? So it will code for phenylalanine, right? For leucine, there are 6 codons, right? UUA, UUG, CUU, CUC, CUA, and CUG. There are 6 codons for leucine. So you can see that for some of the amino acids there are multiple codons are possible, right? Which can, there are multiple codons which can code for the same amino acid. For example, for leucine, there are 6 codons, right, possible, which can code the leucine, right? But the point is that each codon, right? For example, anything CCU or CCCUU, CUU will always code only for leucine, not other amino acids. CCU will always code for proline, but not for other amino acids. But the vice versa is not true. Leucine can be coded by 6 different codons, right? Serine can be coded by 4 different codons, right? And right, same for proline. Proline also can be coded by six different codons, right? But for and this UAA, UAG, and UGA, these 3 are the stop codon, right? They are the stop codon. So when ribosome, so when your mRNA will have this sequence in the mRNA, which are the stop codon, then the translation process will come to the end. So that we will discuss. And tryptophan is an example of a amino acid which has UGG as the codon, and it has only 1 codon, right? Not more than one. And for example, methionine also, if you see the methionine, AUG, which is generally the start codon, right? AUG is a start codon, and that codes for the methionine. So methionine will only be coded by 1 codon, which is AUG, right? So methionine and tryptophan, they are amino acids which are coded by single codon. So here you can see from this chart that met and tryp has 1 number of codon, but aspargine, aspartate, cysteine, glutamine, glutamic acid, histidine, lysine, phenylalanine, tyrosine, all of them have 2 amino acids, 2 codons, right? Isoleucine has 3 codons, AUU, AUC, and AUA, right? Alanine, glycine, proline, threonine, valine, arginine, threonine, and valine, they have 4 codons, and leucine, which we saw that leucine has 6 codons, arginine has 6 codons, right? And serine also has, arginine has C 1, 2, 3, 4, and so 1, 2, 3, 4, and these 6 codons, and serine also has 6 codons, 2 here, AGU, AGC, and right, 2 here, and apart from that, here UCU and UCC, UCA, and UCG, this 4 and this 2, there are 6 codons for serine. So histidine, lysine, and so arginine, leucine, and serine, they have 6 codons, right? So this is called the degeneracy of the genetic code, where except mate and tryp, the other amino acids can be coded by multiple codons, right? So this is called the degeneration of the genetic code, when the one amino acid can be coded by multiple number of codons. Right. So now we will talk about the tRNA. So tRNA, this is the tRNA, and which will, as you know, that which will carry the amino acids. So this tRNA will interact with the mRNA, right? So this anticodon loop, and mRNA has the codon. So suppose, for example, this codon AUC, which is a codon for, if you see here, U, here if you see that A, U and C, which is isoleucine. So this will code for isoleucine, right? This is code for isoleucine. So this amino acid will carry isoleucine and this will interact to this codon by its anticodon loop, which at this anticodon loop of the tRNA. So that's why it is called as an adapter. This anticodon loop has the complement amino acid, complement nucleotide of this mRNA. For example, if it is AUC, then it will be from 5 prime to 3 prime, it will be G, A and U, which is complement, right? So that's how they can make hydrogen bond and they can interact, and which will carry the amino acid here that we will see. So that is called pairing of tRNA with the mRNA, anticodon and codon pairing by hydrogen bonding. For example, here for the codon CGA, which is from 5 prime to 3 prime, if it is CGA, then from the 5 prime to 3 prime in the anticodon loop, it will be ICG, right? And if the codon is CGU, then in the anticodon loop, it will be I, C and G, right? Here I is the inosine. Inosine can make a pairing with uracil and adenine and cytosine, right? So that is called the pairing of tRNA. Now you will see that how this pairing of tRNA happens. That is a hypothesis called Obel hypothesis. So Crick proposed a set of 4 relationship which is called as Obel hypothesis. So the first hypothesis, which is stating that the first 2 bases of the mRNA codon always form strong Watson-Crick base pair with the corresponding bases of the tRNA anticodon, right? So first 2 bases of the mRNA. So first 2 bases of the mRNA will pair with the corresponding bases of the tRNA. So this 1 and 2 nucleotides, they will make the base pairing, right, with the 2 and 3 of the anticodon nucleotide, right? So 1 and 2 will interact with 3 and 2 respectively. 1 will interact with 3 and 2 will interact with 2, right? So these 2 are strong, so they will right, and they are the most specific. Now right, so now when for example here inosine is the first Ubal nucleotide in the anticodon, right? For example, if the codon is CGA, right, then first 2 is the CNG in the codon, so that will base pair with the 2 and 3 of the anticodon, right? Fine, but the third nucleotide in the codon, which will base pair with the first nucleotide of this anticodon, that is inosine, right? So this is a Ubal position, or which is a Ubal amino acid, right? So here inosine is the Ubal nucleotide in the anticodon, right, which can recognize 3 nucleotide, right? For example, this here inosine is in the anticodon loop, the same, right, the same anticodon loop. Because of the inosine, it can detect A, U and C. So now in the anticodon, G and C is the same. So of course in the codon C and G will be the same. So 3, 2 and 1, 2 will be same. 3, 2 of anticodon and 1, 2 of the codon will be same. But as the anticodon has a inosine here in the first position or in the verbal position, so which can interact or base pair with A, U and C, right? So this anticodon can actually recognize CGA, CGU and CGC. So CG is the common, but in the third position, or right, so in the third position of the codon, A, U and C can fit. So by using the same anticodon loop of the same tRNA, you can detect 3 different codons, because the Ubal nucleotide in the tRNA anticodon, which is inosine, that can base pair with 3 different nucleotides, right? So that is the reason, one of the reason of the codon degeneracy, where the same tRNA can recognize three different codons, and as the tRNA is the same, so it will carry the same amino acids, right? So for these three different codons, it will encode for same amino acid as the tRNA is same, right? So the first base of the anticodon, which is the vowel, that will pairs with the third base of the codon, right here. So third base is A. The first base is for the anticodon is inosine, right? And when the first base of the anticodon is C or A, the base pairing is specific, right? So this base pairing G and C. For example, here the second and third nucleotide of the anticodon and first and two nucleotide of the codon, this interaction is specific, right? Completely specific. But because they are recognizing only one codon, but for the vowel nucleotide it can recognize three, right? So that's why it is not that much specific, right? So it is because it is recognizing A, U and C. So this is not specific, right? So when an amino acid is it's specified by several different codons, the codon that different either of the first two bases require different tRNA. So if there is any so suppose an amino acid is specified by several different codons, which we saw in the codon chart, then the codons that differ in either of the first two bases require different tRNA. So if for example here all of these codons are different, but the difference lies only in the third nucleotide, which is pairing with the vowel nucleotide, right, in the anticodon. So that's why tRNA is same, right? Because second and third nucleotide and first and two nucleotide of the codon are same. I mean, yeah, right. So here the differences in the codon is the third nucleotide, which is pairing with the vowel nucleotide, right? But the first two nucleotides are same, CG, right? So that's why they can interact with the same tRNA. See, right? Because right. So this three different nucleotide. So the nucleotide difference in the third position that can interact with the same amino acid, same anticodon, right? But if there is any difference in this first or two position of the codon, right, then they will require different tRNA, right? If only the difference is in the third codon, the third nucleotide of the codon, which can base pair with the first nucleotide or the vowel nucleotide of the anticodon, then you can use the same tRNA. But if the difference is between these first two nucleotides, then it will require different tRNA. Right? For example, here in this example, you can see that in the codon suppose YXG and this codon is YXU. So the first two nucleotides are same. So only the changes in the third nucleotide, which base pair with the vowel nucleotide of the anticodon, right? But this so for that you will require two different tRNA for that, right? Because the vowel position has although the difference is only in the vowel position, but still this C can only detect G, it cannot detect U, right? Similarly, this A can only detect U, not detect G, right? So in this case, this codon can only be detected by this anticodon, not this one. So for this case, for two different codon, you will need two different tRNA, right? And for example, here this anticodon XYU, so it can interact. I mean, it can recognize this. U can recognize A or G. So the codon YXA and YXG, both of these codon YXA and YXG, they can be recognized by this same tRNA. Right? For example, XYG, G can recognize C and U. So YXC and YXU, these two different codons can be recognized by the same tRNA. And for example, here we saw that XYI is the right. So if the I is the verbal nucleotide, that can recognize A, U and G. So YXA, YXU, YXC, right? So all three codons can be recognized by this same anticodon, right? Same tRNA. So this tRNA can recognize three different codons, right? So that this verbal, so that's why this verbal base of the anticodon determines the number of codon a tRNA can recognize. If the verbal position has C or A, then they can detect only one nucleotide, right? Right in the codon. So yeah, so for that, for this codon, right, you will get only one anticodon, right? Only C can recognize. But if you have A and G, so that can be recognized by the XYU. So if the verbal position is U, then it can recognize A and G. If the if the verbal position is A, it can recognize only U. Right? So for this, so for this thing you can need the only one tRNA. But if the vowel position has inosine, then it can recognize YXA, YXU, YXC, 3. So one anticodon can recognize three different codons, which is not possible for here. This anticodon, because it has C in the vowel position, it can recognize only this YXG codon, not others. Similarly here, when the A is in the vowel position, then it can only detect the codon YXU, right? Not others. And as we know that this if two different codons has the any difference in this first two nucleotide, then it will require the different tRNAs. Whatever the vowel position is, it is independent of the vowel position. But if any difference in this first two nucleotide, it will definitely require the different tRNA. But if the first two nucleotides are same, then now it depends what is the nucleotide in the vowel position, right? For example, here C and A, they can only detect, C can detect only G, A can detect only U. So despite of the no change in the two codons, the two nucleotides, first two nucleotides of the codon, right? It will still require only these two codons require two different tRNA, although the first two positions are same. Why? Because the vowel nucleotide in the anticodon can recognize only this, right? So for these two, YXC can be recognized by C and YXU can be recognized by XYE. So both of them will have different tRNA. But if the see here also in the codon first two are same, and if there is A and G, right, YXA and YXG, they have the same first two position included, only the third position is different, then it can read only one tRNA, right? Because A and G can be detected by the tRNA which has U in the vowel position. Similarly, YXC, YXU, they have the same two, first two nucleotides are the same, but still they require only one tRNA to detect, because this C and because this G, which is in the vowel position, that can base pair with C or U both. So those two can be recognized by the same tRNA, and here when there is YX, A, U and C, then they will require only one tRNA, because this XYI, when I is in the vowel position, then it can detect or it can base pair all three of the codons, right? So these three codons, they require only one tRNA which has XYI. So that's why the verbal base, first thing is, if there is any difference in the first two nucleotide, it will require different tRNA. And now also the composition of the verbal base, that will verbal base of the anticodon, that will determine the number of codons a tRNA can recognize, right? If you have any doubt, you can ask. Now next we will study the ribosome. Ribosome, as we discussed, where the protein synthesis takes place. So this is bacterial ribosome, 70S, and this is eukaryotic ribosome, which is 80S. So the 70S ribosome in the bacteria, it is composed with uh, a 5S ribosomal RNA, which we discussed in the last class. The 23S ribosomal RNA in the large subunit, and in the small ribosomal subunit there is 16S ribosomal RNA. Right, for the eukaryotic it has 6S, and we saw in the last class that this for bacteria the 5S, 16S and 23S, they 23S, 5S and 16X, they are in the same primary transcript, right? But for eukaryote it has 80S, so the larger subunit is 60S, so which has 5S ribosomal RNA, 28S ribosomal RNA and 5.8S ribosomal RNA, and the small subunit it has 18S ribosomal RNA. So in the primary transcript this 28S ribosomal RNA and 18S ribosomal RNAs are there in the eukaryotic ribosome, right? And now if we talk about tRNA, we actually studied the tRNA in the transcription process. We saw that tRNA can undergo some modification, for example trimming by RNSP, and then there is some modification in the bases. For example, there is a methylation in the guanosine we saw, right? M1G, M2G, then psi, which is actually pseudo uridylation, which we saw, right? So there is a modification and there is a cleavage in the 5' end. This entire chunk is removed, and in the cleavage in the 3' end this chunk is removed, and there is addition of this CCA in the 3' end, so which is a site for amino acid attachment. Right, so it has so this is the structure of the tRNA. So it has a D arm, right? D arm, where, right, D arm, there is a anticodon loop, and there is a, right, and there is a amino acid arm. This is the amino acid arm, where the amino acids are getting attached, and there is a T5C arm. This is the T5C arm. Here you can see the T and phi, that's why it is T5C arm. This one is D arm because that is the presence of D, and this is the anticodon loop, right? So this is the 3-dimensional structure of the East tRNA for phenylalanine from X-ray diffraction data. So now for the translation, tRNAs have to be charged, right? So this charging is called the amino acylation of tRNA, right? So how does it takes place? So suppose there is a amino acid here, right, and this is the ATP. So this amino acid has the C double O minus, so O minus is electronegative, so this will make a nucleophilic attack to the alpha phosphate of ATP, right? So this bond will be broken, so beta gamma phosphate will be removed as a PPI, and this this AMP will be conjugated with the amino acid, right? This is called charging of amino acid, or right. So this is actually amino acyl AMP, right? So this is called 5' amino acyl AMP, where AMP is charged with the amino acid. Now it has to be conjugated with the tRNA, right? So this amino acyl tRNA synthetase will catalyze it. So for suppose this is a tRNA which has a CCA at the 3' end. So at the last, nucleotide is the adenosine, right? So this is your adenosine, right, which is in a form of AMP. So now this AMP, it has a ribose sugar, and this 2' OH, right? This oxygen is electronegative, so it will make a nucleophilic attack to this amino acyl AMP, right? So here this phosphate is electro deficient, so yeah, and this carbon as well. So this oxygen will make a nucleophilic attack to this carbon, right? So this bond will be broken. So AMP will be released here. AMP will be released, and this amino acid will be charged to the 2' OH position here. So the entire amino acid is charged with the 2' OH region of this uh, tRNA, which has adenosine as the at the 3' end, right? So after that, trans-esterification will take place, so that this bond will be broken, and this 3' OH it also has the oxygen which is electronegative, so that will make a nucleophilic attack to this electron deficient carbon, right? So this bond will be broken, and this amino acid will come here, right, to this 3' OH position, and then again there will be regeneration of the 2' OH, right? So this is called the amino acid tRNA. This is one pathway. Another pathway is from here. If there is a class 2 amino, this is for class 1 amino acid transferase, and if the enzyme is the class 2 amino acid transferase, then it will conjugate the amino acid from the amino acid AMP to the 3' OH group. So again, this is the tRNA. At the 3' end there is adenosine. So the 3' OH, which has oxygen electronegative, it will make a nucleophilic attack to this electron deficient carbon, right? So this bond will be broken. So AMP will be removed, and the amino acid will be conjugated to the 3' OH position, right? So these are the two different pathways. So for class 1 amino acid tRNA synthesis, it will take 2 steps, including the trans-esterification. For class 2 amino acid tRNA synthesis, there will be only 1 step. So now the amino acid is conjugated with the tRNA, which is called amino acid tRNA, right? So that is called the charging of the amino acid, right? So then now tRNA will carry this amino acid, and it will incorporate to the polypeptide chain, right? So this is the structure of the amino acid tRNA, where the tRNA is there, T form, T5C arm, anticodon loop, and this is amino acid arm. So at the 3' end there is CCA, means adenosine. So in the ribose sugar of the adenosine, in the 3' OH position, this amino acid will be conjugated, right? So this is called the so this is the structure of the amino acid tRNA. Now the structural element for tRNA that is required for the recognition by tRNA synthetase. So that is your amino acid tRNA synthetase, which will conjugate amino acid to the tRNA. So, and this tRNAs are specific. Particular amino acids will be recognized by a particular tRNA only, right, which is specific for this amino acid. One amino acid, there is only one tRNA. So, suppose for example, this is the tRNA for alanine, right, which can carry the alanine amino acid. So it has in the amino acid arm there is a GU, right? So this GU, which will make a double, two hydrogen bond, right? So this is the only element which is required for the specific binding to the enzyme and the amino acylation, right? And the proof is when a short synthetic RNA mini helix, which has a critical GU base pair, right, but lacking most of the tRNA structure, right? So this structure is lacking most of this tRNA structure, but it has a G11U base pair. But this is also able to so this is also able to carry the alanine by the alanine tRNA synthesis, right? So so this GU base pair for alanine, for the recognition of the alanine tRNA synthesis. So this double bond, this is only playing a crucial role. This is enough so that this tRNA can be recognized by the alanine tRNA synthesis, and it will be charged by a alanine amino acid, right? So now we will see we will see the initiation of the translation in bacteria. We will see. Right. So here, first the two subunit of the ribosome, 30S and 50S, will be separated. Right. So first, this initiation factor 1 and 3 will come, and they will bind to the P site of the 30S ribosomal subunit. Right. So and then it will recruit the mRNA, right? And this mRNA will position to the 30S ribosomal subunit by this way that when it will interact with the shine Delgado sequence of the ribosomal RNA, right? Sorry, the the mRNA will have the shine Delgado sequence which will interact with the complementary sequence in the ribosomal RNA in the 30S subunit. So that we will see later. Right. Here you can see that this is the shine Delgado sequence which is present in the prokaryotic mRNA, which is AGG-AGGU, and it can interact with the 16S ribosomal RNA, which is a part of this 30S ribosomal subunit. So it will interact with the 16S ribosomal RNA, which is just complementary, right? So that's how they can. So this mRNA can position themselves in the ribosome where it can interact with this with the 16S ribosomal RNA by this shine Delgado sequence or SD sequence, right? So yeah. So now first mRNA will align them to the ribosome, right? Where their shine Delgado sequence will interact with the corresponding complementary sequence in the 16S ribosomal RNA, and then this AUG will be fitted in the P site of this 30S ribosomal subunit, correct? So yeah, so that is the initiation codon or start codon. So now the start codon, that is code for the methionine. So now in the next step, the IF2, which is correct, which is bound with the GTP. So IF2 GTP will recruit this tRNA, amino acyl tRNA, which has that UAC, the anticodon loop, and it is carrying the F methionine. So this amino acyl tRNA will be recruited into the P site where this initiation codon is there, and as the anticodon has the UAC just complementary, then that's why they can interact with this start codon in the mRNA, right? So along with that, F methionine will be there, and then an IF2 GTP will also be bound here. The GTP hydrolysis will be taken place. So from IF2 GTP will be released as a GDP and PI. Then IF1, 2 and 3, both of them will be released. Then at the last step, the 50th ribosomal subunit will come and it will bind, right? So now the translucent process will be initiated, right? So here initially when IF1 and IF3 are bound with the 30th ribosomal subunit, so that will inhibit the association of the 50th ribosomal subunit, right? It will only allow when the IF2 GTP will recruit this amino acyl tRNA of Fmate in the start codon, right? Then only when this GTP will be hydrolyzed and then IF123 will be released, that will allow the 50th ribosomal subunit to bind with this mRNA and mRNA and 30th ribosomal subunit, where the already in the start codon there is amino acyl tRNA which is Fmet is there. So this is the initiation complex in the bacteria. So right. So now the translation has been initiated in the bacteria. Now we will see how the elongation steps take place. Right. So this is what we talked about, shine Delgado sequence, right? So which can pair with the 16S ribosomal RNA. The different gene of the E. coli or the mRNA of the E. coli, what Trypae, Arabi, Laci, right? So those when they has to be expressed in a form of protein, then they have this different shine Delgado sequence, right? And this is the prokaryotic mRNA which with consensus shine Delgado sequence, which is AGGU, which can recognize or base pair with the 3' with the 16S ribosomal RNA, right? Which is complementary, right? So that's how the mRNA can position itself to the 16 to the 30S ribosomal subunit, right? And the stop codon AUG will be just very close to the shine Delgado sequence. So that is the translation start site. So here actually, which is the start codon, here actually this F-mate tRNA along with amino acid will come and bind, right? So this portion will be positioned in the P site AUG. If it can recognize the shine Delgado through the shine Delgado sequence to the 16S ribosomal RNA, then they can be able to fit this AUG or start codon to the P site of the ribosome, right? And then only the F-mate amino acid tRNA will come and it will be recruited to this P site where the start codon is there, right? So next we will see how the elongation takes place after initiation of the translation. So this was for prokaryotic initiation, but for eukaryotic for eukaryotic the things is just quite a bit different. So as you know that in the eukaryotic mRNA there are the processing of the eukaryotic mRNA. So one of them is the 5' G capping, splicing of the introns, and there is poly-A tail addition, right? This is your eukaryotic matured messenger RNA which has a 3' poly-A tail. So this poly-A tail can bind to the poly-A binding protein or PABP here, right? Which can interact with the EIF4G. The EIF is eukaryotic initiation factor. So EIF4G, which can interact with the EIF4E, right? Which can interact with the 5' cap of the mRNA. So this EIF4G and EIF4E are the part of the larger complex called EIF4F. So this complex can bind to the 40S ribosomal subunit. So through this EIF4E and 4G, the mRNA can bind to the ribosome because this is as a mediator, right? And so 5' cap of the mRNA is binding with this EIF4E, and poly-A binding protein is actually connecting this 3' poly-A tail of the mRNA to the EIF4G, right? And this EIF4G and 4E, which are part of EIF4F, they are binding to the 40S ribosomal subunit, right? So that's how through this EIF4F complex, the mRNA of the eukaryotic they are also binding with to the 40S ribosomal subunit, right? So this is the eukaryotic initiation. Now the elongation. So as we saw that for initiation of the eukaryotic in the P site, the translation start site or the start code will be placed so that the F-mate, which is the first amino acid along with the tRNA will come and bind, right? After that, elongation step will be happening. So this is the last thing we saw, that in the P site, the amino acid tRNA F-mate, which is the first amino acid, is bind to the P site. Then in the elongation step, the next amino acid tRNA will come, right? So the next amino acid tRNA, which is this one, the amino acid AA2, suppose. So that will come to the bind to the A site of the ribosome, right? And it will require associated factor, which is TU, right? So TU GTP, right? Which is the TU is the elongation factor. So this incoming amino acid tRNA and TU-GTP, that will make this complex amino acid tRNA along with this TU-GTP, right? And then it will bind to this A site, right? When this GTP will be hydrolyzed, so TU-GTP and phosphate will be released, and this amino acid tRNA will be bind to the A site of the ribosome, right? And then again, TS will change this, exchange with this GTP with GTP, right? So again it will be T from TU GTP, it will be TU GTP. Then again it can take part for the next part of the cycle, when it will take the new amino acid tRNA, which suppose that is AA3, and it will make this amino acid tRNA TU complex. Then again GTP will be hydrolyzed, and this next amino acid will be placed into the A site, right? Here also the A2 amino acid tRNA will be placed into the A site, which is followed by the removal of this TU, right, by the GTP hydrolysis, and this, and after that this TU GTP is exchanged with the GTP by TUS, right? So now TU is TU GTP, then again it will take part for the next incoming amino acid tRNA. So now this next amino acid tRNA AAT is binding with the A site of the 30S ribosomal subunit, right? Right. So now in the next step there will be a so now in the A site the 8 amino acid tRNA A2 is there, and in the P site the amino acid tRNA F mate is there, right? Which was formed through the initiation step. So now in the amino acid tRNA F mate, this, and as we know that the binding of the amino acid to the tRNA by amino acid tRNA synthesis, so that we saw here that the binding is with the 3' OH of the adenosine of the 3' end of tRNA, that will be bind with this amino acid. So this carbonyl group, right? O C double bond O, right? This is which is a ester. So if you will remember that, then you will see that this is the case, right? Which tRNA is binding to the carboxylic acid group of the amino acid, right? Right. And it has a so now, and the next amino acid, so it has the lone pair of electron in the nitrogen in the amino group. So this will attack, so this will make a nucleophilic attack to this electron deficient carbon here. So this bond between amino acid and the tRNA will be broken. So this so there will be formation of the peptide bond, where this nitrogen will make a nucleophilic attack to this carboxylic acid end of this F mate amino acid, right? So now there will be a bond between this C double bond O and amino terminal of the AA2, right? So yeah, so this is the AA2 and this is the F mate, right? And there is a bond between the amino terminal of the AA2 and the carboxylic acid of the first amino acid, which is F mate, right? So now this amino acid is transferred from this tRNA, right, of the P side to this chain, right, which is the tRNA of A side, right? So now this tRNA will be empty. So now the next step is the translocation. So now the ribosome will move in this direction, right? So that this so ribosome will move to this direction from the 5' to 3', right? So as a result, as it will move, then this E side, so see, till now in the A side these two amino acids are conjugated with this amino acid tRNA, right? And in the P side the tRNA is empty. And now if the ribosome will translocate to 5' to 3' direction, then this E then this empty tRNA, which was initially at the P site, that will come into the E site, and then the A site will be empty, and this dipeptidyl tRNA A2, that will be placed in the P site, right? See, the same thing has been happened, that after this, so this was initial complex where the dipeptidyl is has been formed, right? So the empty tRNA was in P site, and dipeptidyl tRNA A2 is in the A site, and E site is empty. And then if the ribosome will translocate through this 5' to 3' direction, right? Then you can see that this, this empty tRNA will be the exit site of the ribosome, right? Right, A site will be empty here, and the right, and the P site, and this dipeptidyl tRNA2 will be placed into the P site after the translocation step. So as I know that from the E side everything will leave the ribosome. So this empty tRNA, as it will come to the E site, it will leave the ribosome. Right. So the ribosome will be left with the empty A site after translocation. It will be left with the empty A site, dipeptidyl tRNA2 in the P site, right, and the exit site is again empty. So now the next incoming amino acid tRNA for the third amino acid, that will come, and that will bind to the A site. Then again this bond will be found uh, between this dipeptidyl chain and the fresh, the incoming amino acid tRNA3. Then it will be again this translocation happens. So for this translocation step, there is the EFG is there. So EFG is binding with the GTP. So then after the GTP hydrolysis, the energy released from the GTP hydrolysis is used for the translocation step in this translational elongation. Right. So EFG is the EFG is the translocase in this case, right, which is helping the ribosome to translocate so that the A site will be empty. This dipeptidyl tRNA2 will be placed in the P site, and this empty tRNA will be placed in the E site. So now the empty tRNA will leave the ribosome as it is in the E site, and the next incoming amino acid tRNA3 will come to the A site, right? Again the same thing will be happening, right? So that's how the elongation step will be taking place in the case of translation, right? So if you have a doubt, you can. now we discuss about the elongation step. So now for termination in the protein synthesis, so when this cycle will start, will keep continuing, the new amino acid tRNA will come to the A site, right, and then translocation. So when the ribosome will reach to the stop codon, for example UAG, right? So when after addition of the last amino acid, when translocation step will be occurred, then A site will be placed, right? Then the A site will be placed to the UAG, which is the stop codon, and P site, right? And the P site will be placed in the polypeptide chain, which is attached with the tRNA, last tRNA. So now, when this UAG will stop codon will reach to the A site, then the release, then there is no amino acid for that. So release factor will come. This release factor for only for this particular stop codons, UAG, UGA, and UAA. So only for the stop codons, when they will be placed into the A site, then the release factor will come and it will bind to the A site of the 30S ribosomal subunit, which will induce the hydrolyzation of this polypeptide tRNA bond, right? Initially it was polypeptide tRNA at the P site, but now upon addition of the binding of this release factor will cause the hydrolyzation of this polypeptide tRNA bond, right? So now this polypeptide chain will be released from this tRNA and from the ribosome, and in the P side there will be empty tRNA. Then when this polypeptide chain will be released and there will be empty tRNA, then this empty tRNA will be also released, right, along with the release factor, right? And in addition, the 30S and the 50S ribosomal subunits will also be dissociated from this mRNA, right? So upon dissociation of this, upon hydrolyzation of this polypeptide tRNA bond, polypeptide chain will be released, and then the empty tRNA release factor and the 30S and 50S ribosomal subunits will be dissociated from the mRNA, right? So that's why. So that's how the termination of the protein synthesis happens. So in the prokaryote, the transcription and translation both are occurring in the cytosol, right? But in the eukaryote, transcription is occurring in the nucleus, and then the mRNA has to be exported from the nucleus to cytosol, where the translation will start occurring, right? But in bacteria, there is no such nuclear membrane, so transcription will also take place in the cytosol, and translation also takes place in the cytosol. So bacteria can couple the transcription and the translation in the cytosol, right? That means simultaneously transcription and translation will be happen. So this is the data duplex. So this is using as a coding strand, right? And this is the direction of the transcription going on, which is synthesizing this mRNA from this template DNA, and then this mRNA is used by the ribosome, right? They are binding to the mRNA, and ribosome is synthesizing protein from the mRNA, right? So here the transcription is going on. Along with that, simultaneously the ribosome is doing the translation also, right? So this is direction of the transcription, this is direction of the translation. So as both of the processes, transcription and translations are occurring at the same time, so that is called the coupling of transcription and translation in the bacteria. Now we will talk about some inhibitor of this translation, right? For example, puromycin. Yeah, so pyrimidine is a translational inhibitor. So it is a it has very much similarity with the amino acid tRNA. So it can be called as the amino acid tRNA analog, right? So when the transcription, so when the translational elongation will be going on, right? Then after the last translocation step, A site will be empty, P site will have the polypeptide tRNA. So now in the A site, this pyrimidine, which is a analog of the amino acid tRNA, so this pyrimidine will come and it will bind to the A site, and then the thing which we know that the in the P site, the amino acid, the carboxylic acid terminal of this amino acid, that will be attacked by this uh, amino terminal of the pyrimidine, right? Nucleophilic atom. So then there will be a formation of this bond, and the bond between the tRNA and this polypeptide chain will be broken. So then, as we know that this entire polypeptide chain will come to this A site by joining with the latest amino acid. Yeah, right. So, but for this case, when this reaction will be happen, this between bond between the amino acid and tRNA will be broken, and this polypeptide chain will be transferred to this amino acid tRNA analog in the A site, which is pyrimidine, right? By forming this bond, right? So after forming this bond, it will become this thing, right? Polypeptide chain followed by the pyrimidine in the A site. At the P site, the tRNA will be empty, right? So that will actually block the further translation process, because now there is no tRNA. Yeah, so as there is no tRNA, right? So that's why this polypeptide chain followed by this pyrimidine. This is the structure actually, polypeptide chain. Then this pyrimidine. So this, as it cannot bind to the A site because it doesn't have any tRNA. So this portion will be, so yeah. So after forming this complex, there will be no translocation. Before translocation, as there is no tRNA, so this polypeptide followed by this pyrimidine will be dissociated from the ribosome, right? Which is causing the premature chain termination, right? This is the structure of this polypeptidine pyrimidine, right? So this polypeptidine pyrimidine will be released from the ribosome, dissociated, and there will be termination of the premature. So this is called a premature chain termination, right? So that's how the translation can be inhibited by using the amino acid tRNA analog, which is pyrimidine, right? So that's all for today's protein metabolism session. If you have any question, you can ask me. Okay, so as there is no further question, so we can close the session. So see you on the next week.